We've done some work looking at a combination of drugs, sapacitabine in combination with seliciclib. These are two agents that we've been testing in patients with advanced solid tumors. Some of our initial work had focused on looking at really dosing schedules of these two agents. The sapacitabine is an oral nucleoside analog, and it causes double-stranded DNA breaks, whereas the seliciclib is a CDK2 and CDK9 inhibitor, and it results in a decrease in anti-apoptotic proteins, and it also diminishes the HR repair machinery.
Could you talk us through some more of the process of the DNA damage from the sapacitabine?
The sapacitabine gets metabolized into this compound called cNDAC. cNDAC gets incorporated into DNA, and it causes initially single-stranded DNA breaks, but then upon further replication, it causes double-stranded breaks. And if these breaks aren't repaired, it results in cell death. So what we found is that if you have a tumor that is deficient in homologous recombination, sapacitabine seems to be a very efficient compound, and it seems that the efficacy is probably enhanced by adding the seliciclib, because that's impairing the repair of the DNA damage that was caused by the sapacitabine.
This is something that was being administered sequentially and concomitant to other therapies. Was there any difference between one first and the other?
We looked at two different dosing schedules. The reason is that there was some preclinical data that suggested if you give sequential therapy, it seems to be synergistic, whereas if you give concurrent therapy, it potentially seemed like it was antagonistic. So we looked at one schedule in which sapacitabine was given twice daily for 7 days, followed by 3 days of seliciclib, followed by 11 days off in a 21-day cycle. And then we looked at a second schedule, which we call an interleaved approach, where it's really alternating dosing. So a patient will get sapacitabine in the morning, and they'll get seliciclib in the evening. And it really is trying to imitate a sequential schedule, because the half-life of these agents is so short that the drug will mostly be washed out by the time you've given the second drug. And so we looked at both of these schedules in this phase 1 study.
And what kind of results have you got from that?
So initially, when we looked at the sequential dosing schedule, we enrolled patients with all solid tumors. And it turned out that the responses were seen only in patients who had a BRCA germline mutation. And so when the first cohort of the sequential schedule was done, the objective response rate was 25% in patients who had a BRCA mutation. And so then we decided to limit enrollment to the second schedule, that interleaved approach, to patients who just had a BRCA mutation. And in that schedule, the objective response rate was 7%. So it was slightly lower than with the first schedule.
Okay, so we're using the marker there, BRCA. It's a very famous mutation. It's one that's entered kind of the common parlance. But do you think it does have a function as a future biomarker for developing therapies?
I think in this particular case, because the sapacitabine's DNA damage is really HR dependent, it seems like a biomarker for response to this agent may in fact be having a germline BRCA mutation. And that's where we're seeing the predominant efficacy, which I think is encouraging, because even in the patients who had a germline BRCA mutation, many of them had progressed on prior platinum-based therapy, and several had even progressed on prior PARP inhibition. So I think these patients were generally heavily pretreated, many of whom were treated with agents that were trying to take advantage of their HR deficiency. So it's encouraging that the regimen seemed to have continued activity.
So I suppose, where next then? You mentioned this was a phase 1 trial. Are there any phase 2 plans?
So we just started enrolling to an expansion cohort to this phase 1 trial, where we're limiting enrollment to patients who have metastatic breast cancer and a germline BRCA mutation. And we're actually mandating biopsies at baseline to really help us understand how the HR machinery is in those patients up front.